Welcome, everybody. I'm going to talk about the last pandemic, the one before that, and this one. If you don't know me, my name is Michael Emerman. I'm a virologist at the Fred Hutch. So my lab studies HIV. We study the evolution and function of HIV, of restriction factors against HIV. We try to understand how did HIV become a human pandemic virus. And we've also started studying HIV latency. I'm not a coronavirus expert, but I do teach MCV 532, human pathogenic viruses. So that's the graduate virology class we teach here every other year, and I've done that since 2006. So this talk is mostly a mixture of lectures that I give in that class, especially the coronavirus lecture, updated with new stuff. And this is not my particular area of expertise, but I know a lot of virology. I taught this class for a long time. I used to be editor-in-chief of virology, so that's what this is based on. What I'm going to do is I'm going to talk about the last pandemic and the first, and the emphasis there is going to be why that pandemic was not searing your brain as being such a horrible thing, because it could have been, and the advantage we had last time we don't have this time. Then I'll start talking about coronavirus biology in general, and we'll get into SARS and now the new pandemic. Just to be clear about nomenclature, so COVID-19 is the disease, SARS coronavirus 2 or SARS-CoV-2 is the virus. I'm going to be mostly talking about the virus. So the disease and the virus are not the same name, similar to the way that HIV is the virus, AIDS is the disease. Most people with HIV do not have AIDS. Most people with SARS-CoV-2 do not have the disease. So we're talking about the virus, and those two things are not exactly synonymous. So let's start with the last pandemic. The last pandemic that we had was the H1N1 flu. It started late in the flu season. So flu season usually ends around March-ish, sometimes a little bit later, but there was a large increase in cases of influenza-like disease in Veracruz in Mexico, which went up and then eventually resolved a month later. It was unusual in having severe illness in young adults. It could not be typed to a known influenza. So it was influenza A, but they could not subtype it. So subtyping means H1 or H3 usually for the influenza viruses in humans. They sent samples to the CDC and to the equivalent in Manitoba, and it was typed as swine flu. By that time, by early April, it also matched 2 cases in San Diego. So it had already spread by then. Its attack rate was usually high, so 28% of this in Veracruz, so 616 out of 2,000. Very high in children, less in adults, and this will be important in the next slides. So this spread very, very rapidly. It was originally called swine origin H1N1. Now it's called H1N1 pandemic. So note these scales in millions. So between April and the next start of flu season, the next October, there were 20 million cases in the United States. The vaccine came too late to help. So vaccines are chosen in the springtime, so February, March. It takes them time to make that vaccine available for the next November, October. Before the next flu season, the vaccine was not ready. If you were at the Hutch 10 years ago, you might remember that the line to get a vaccine was going out the doors to the elevator. Everyone in that line was getting the wrong vaccine. The right vaccine was not available until November-ish, by time most of this was over. So then next season, October through the next November, so these are cumulative infections now, it jumped from 20 million to nearly 50 million. So this is what happened October to November of the next season. It continued to climb somewhat, but basically leveled up after that at about 70 million people infected. It didn't peak until May, June. So we will talk about in another slide about the seasonality of respiratory viruses, including coronaviruses, but it's not an absolute. So transmission goes down in the summer months in the Northern Hemisphere. That is mostly correlated with relative humidity. So how much moisture the air can hold. When it's cold, the moisture holds less air. But it's not an absolute. It reduces transmission, but doesn't make them go down to zero. This one peaked in May, June. So the WHO actually declared this pandemic the first week of June. It went down in July, August, and it came back in October. So the idea that in the current pandemic with this coronavirus, that heat will kill it, that's completely untrue. It's going to make transmission less efficient, but it's not going to go away exactly. If it is not controlled, meaning not controlled in the Southern Hemisphere, during their winter, it'll be back in the fall. With this, what's called often the second wave. So second waves of respiratory viruses, flu viruses especially, are going to occur in October. So respiratory viruses are seasonal. It's well known for influenza. So this is just one particular study, but this study is nice because they also looked at human coronaviruses. So we'll get to in another slide or two. There are human coronaviruses. Let me minimize that window. Okay. There are human coronaviruses, and they are also seasonal. So here is for some reason that's not, okay. People see the cursor? Can someone type yes if you can see the cursor? Okay, great, I'll use the cursor then. So influenza goes up, it goes down, it goes up. So this is winter, summer, winter, summer, winter. Under here are the things which are less common than influenza, but the human coronaviruses, these three here, OC43, 229E especially, and they're following pretty much the same pattern. So up, down, up, down, up. But you can see they're not completely going away in the summer. It's just being less frequent. So with this one, with this particular coronavirus, we would probably expect to see the same thing. This transmission will be less efficient in the summer. Again, the social distancing and the reduced transmission in the summer is hopefully going to have an effect on what's going to happen in the next fall. So why was this pandemic not something that you remember as being the catastrophe that it could have happened? So again, we had 70 million people infected in the United States. In a pandemic influenza year, 30% of the human population can be infected. And that's what happened the last time. That's what happened in 1957 when there was another human pandemic virus, the H2N2, entered the human population. In 1957, there were 100,000 deaths in the US, over 4 million worldwide due to that. That didn't happen with this pandemic, even though as many people were infected. And that's because of something which was incredibly lucky. So there's some history, there's some luck, and there's some biology. It is that it was unusual for its lack of infection and lack of morbidity in people over 70 years old. So the people who would usually die of influenza were not dying of influenza. Here we have a graph of influenza deaths. This is average deaths per month. And these dotted things are the seasons for influenza in the previous 10 years. So you can see there's a little blip here for kids under 2 years old, and then increases with every decade of age. People over 85 especially are susceptible to really serious diseases by influenza. So this is the season. But in red here, this is what happened in the 2009 pandemic, where actually there were fewer deaths than you would have expected. There were also fewer infections in this age group. So here we're seeing the number of infections with 2009 H1N1, so that's the dark purple. And you can see there are actually fewer infections in people who are over 53 than people who are under 53, as opposed to the seasonal influenzas here, where infections, the number of infections are actually pretty constant across the ages. So more people were infected than in a normal flu year, but there were actually fewer deaths than in a normal flu year, only 15,000 as compared to this, and especially where it could have been up across the graph. The reason that this happened is that people who were over 70 years old had antibodies against a virus that they were infected with when they were young. So here's a graph showing that. So here they infected mice with pre-1978 viruses, and then they asked, can those mice make neutralizing antibodies to the pandemic H1N1? So here we infect mice with a virus from 1947, with a virus from 1957, with a virus from 43, and we see these neutralizing antibodies, higher titers neutralizing antibodies, and not neutralizing antibodies against H1N1 if those mice were infected with a virus from 1983 or the year before in 1999, a decade earlier, 1999. Similarly, although with less data, if we look at antibody titers in just serum from people of a particular decade, so someone born in 1930, 1940, 1920, so these people are going to be the people who would usually die in 2010 with this virus. They have neutralizing antibodies against the H1N1, whereas people born younger did not. So the neutralizing epitopes on the pandemic H1N1 were similar to a virus that had previously circulated in the human population. In 1918, H1N1 went into humans and pigs at about the same time. It might have gone birds to humans to pigs, or it might have gone birds to pigs and humans at the same time, or it might have gone birds to pigs to humans. But pigs and humans were infected at the same time. That virus had been evolving independently in humans and in pigs for different amounts, for those amounts of times. Different things happened in humans, but that same virus was evolving in pigs. So the virus that had been evolving in pigs that then eventually contributed a segment to the thing that became the H1N1 pandemic was similar to a virus that had been circulated in humans in the 1940s and 1950s. So this is why we got lucky. it was a pandemic, but the people who would have died did not die. The reason this is important for the current ones is the current one, this will not happen. There is not pre-existing human immunity to this new coronavirus. So this historical thing that happened where the people who would have died did not die, we are not seeing that here because there is not pre-existing immunity in the human population. Any questions about that till I get to the next part about coronaviruses? Okay, I don't see anybody typing any questions. Hope that means that you can still hear me. Let me type a question so I know that you're listening. Okay. Everybody still here? But nobody has any questions. You got to ask a question. Okay, good. I got a question. Then I'll go on. So the question is, you said there are some coronaviruses that are already circulating in humans. Are these usually not so fatal? Yes, we're going to get to that. There are 5 coronaviruses in the human population. Sorry, there are 4 currently circulating. Those are not fatal. Those are causing minor respiratory diseases like the common cold. That's going to be important for what we might expect is going to happen here. Dave Rabel, hi Dave. Was that why the 1918 flu also infected younger people? Yes, that's a great question. And yes, there is a great hypothesis about that. So the hypothesis is that there was a virus circulating around the time of the American Civil War. There's historical evidence that there was an influenza pandemic around the Civil War time. And there is speculation that that was a H1N1 that protected older people in 1918. However, in 1890, there was completely different influenza circulating. And because something called original antigenic sin, that is, you make the best responses against the first thing you saw, people who were in their 20s and 30s were first infected with something, with a different influenza between 1890 and 1918. So they actually were especially susceptible to a new H1N1. Can you determine who is resistant to coronavirus by measuring serum antibody? At the present time, no one has immunity except people who have been infected with this one so far. And it is likely there will be neutralizing antibodies, but it's way too soon to know what levels you need. Okay, there are other questions about coronaviruses, and I think we're going to go on because a lot of those are going to be asked, but thank you for asking questions. So, the viruses we're going to talk about are in the order of nidoviruses. In the order of nidoviruses, there are 3 families. Of those 2 families, one is the coronaviruses. Humans only have coronaviruses of those nidoviruses. The other nidovirus families, there's a lot of viruses of invertebrates. The other family is very important for a lot of agricultural species. So turkeys, pigs especially, there are a lot of those. Coronavirus family is a very large family. Almost every vertebrate has coronaviruses. SARS coronavirus is going to be the one we're going to talk about, so SARS-1 and SARS-2. Nido means nested. You're going to see why that is occurring because that refers to the way the mRNAs are a nested set. Corona refers to crown, and you can see that on these spikes coming out of the virions that have this crown shape. If you want to read a book about cross-species transmissions of virus, Spillover is a really good book, especially the chapter on SARS is one of the best chapters in that book. Here's a schematic of the coronavirus virion. And it has 3 glycoproteins. Spike protein is the one that is the receptor binding, and this is what is responsible for that crown shaped. It's also responsible for fusion, so similar to many other envelope proteins of RNA viruses. The receptor binding, the fusion protein are the same thing, but there's a cleavage event to activate fusion. So spike protein is the thing that the neutralizing antibodies are going to be made to. It's going to prevent binding of the virus to a receptor. Envelope protein here is clade specific. So this can distinguish one clade of coronavirus from another clade of coronavirus. In the diagnostic tests for this coronavirus, so some of the primers are actually in the envelope, so it distinguishes the clade of coronavirus that this is from the common coronaviruses. The nucleocapsid is an RNA binding protein that's going to coat the genome. This is conserved across coronaviruses, so primers and nucleocapsid are going to detect all coronaviruses, or most coronaviruses. And inside the virion, there is a large positive strand RNA. The genome for this virus is RNA, positive strand, so the same sense as translation. There are other accessory proteins that are likely packaged in here that aren't shown. There are no viral enzymes inside the virion. Let me get to that. So that's important because for the stability of the virus. So let me get back here again. This is an envelope virus. So it has it gets part of the cell membrane when it buds out, and these proteins are into the membrane. So that's what makes it susceptible to things you are using to get rid of off your hands. So Purell is alcohol-based. Alcohol dissolves the lipid bilayer of this virus, so it inactivates it by getting rid of that. Soap works, so soap is also going to deal with the lipid bilayer here. Soap has the additional advantage of physically getting rid of viruses off surfaces. If you have a choice between alcohol and soap, take soap. Both of those work. Bleach also works, so bleach is also affecting both denaturing the proteins and denaturing things in the lipids. So that's what accounts for inactivating the viruses on surfaces, like your hands and other things. In fact, there's no enzymes in the virus. So this is a positive strain RNA virus, as opposed to negative strain RNA viruses like influenza. So negative strain RNA viruses have to put enzymes into the virion to get from negative strand to positive strand. In this case, the positive strand is going to be used itself as a message. So that accounts why there's greater stability of the coronaviruses than there are for things like influenza, because they are more stable because the enzymes inside don't get denatured. There are coronaviruses which are strictly gastrointestinal, especially ones studied in pigs. This one also can be gastrointestinal, so they can go through the gastrointestinal system, which kind of indicates they have increased stability versus other envelope negative strain RNA viruses which don't do that. There have been studies on the stability of this particular coronavirus. So in the laboratory setting anyway, it can survive on the half-life on surfaces is about 4 hours, and the detection level on surfaces is about 2 days. In the air, it has a half-life of about 2 hours, but a detection limit of about a day. So relatively more stable than some of the other respiratory viruses. So here are some of the other human coronaviruses, and don't pay too much attention to this tree because I'm going to show you some better trees later. But these two, OC43 and 229E, are very common coronaviruses of humans. They are responsible for, depending on the year, 15 to 20% of common colds. They are upper respiratory tract, whereas the SARS coronaviruses that we're going to be worried about, they're going to be lower respiratory tract. Upper respiratory tract, like the common colds, fewer symptoms. It's what causing your runny nose, more easily transmitted from the upper respiratory tract than the lower respiratory tract, but upper respiratory tract is less susceptible to the damage to the upper respiratory tract is going to be less deadly than damage to the lower respiratory tract. So these are the ones that I showed you on the previous slide, which are seasonal. We see different ones, we see different variants, but these are common in the human population. Most children will be infected with these. People are infected with these in childhood. So most children will be positive by the time they leave childhood. SARS is over here. There are two others. So NL63, also a lower respiratory tract like SARS, but not as deadly. It's responsible for about 2 to 5% of croup in children. So this horrible sounding cough that they get. Less severe, this one is less severe in children than it is in adults, similar to what we're going to see with the SARS-2 coronavirus. And one which is less common over here, this coronavirus. All of the coronaviruses are cross-species transmissions originally. So most of them have a reservoir in bats. So these top four, are now endemic in the human population. That is, they had a reservoir. This one clearly goes back to bats. This one, 229, interestingly, has a reservoir in camels. We're going to see that for this one, MERS as well. Its original reservoir is in bats. OC43 is a super cool one because it has a very closely related relatives in cows. It seems to have transferred several times from cows to people. And when it got to people, now those are now recombining with each other, which are giving different genotypes. It's now endemic in the human population. So it originally came from cows. It is now a human virus. This one, we don't know yet. SARS coronavirus, the first one, clearly has a bat reservoir. We'll get to this one when we get a little bit further. There may be an intermediate host of a civet. It may have just come directly from bats. And MERS, Middle East Respiratory Syndrome, is now endemic in camels. So there is a bat reservoir that went to camels. It's endemic in camels. The transmissions are now from camels to humans, but it is not endemic in the human population. Neither is SARS either. Questions? Brianna, I read a headline, the sequencing Singapore revealed that those under 30 are completely asymptomatic. Yes, we're going to talk about that later on. So that is something that is unusual with coronaviruses, is that not under 30, under 9 seem to be mostly asymptomatic. And the coronaviruses in general, that seems to be their biological property. If they are much less severe in young children than they are in adults. It's not clear what the biology around that is about, but that seems to be the way the coronaviruses keep themselves in the population, by infection of children. With the other coronaviruses, immunity seems to last a couple years anyway. For the ones the common ones, the people will get reinfected as adults, but the reinfection, the symptoms are less severe than they would be for a primary infection. That is something that may happen with this one, which we'll talk about. Frank, considering coronavirus 19 originally in a bat reservoir, okay, the virologist in me can't let this slide. COVID-19 is the disease, not the virus. The virus is SARS coronavirus 2. Anyway, originally in a bat reservoir with limited geographical range, what are the potential consequences of entering other reservoirs? That's a super dangerous thing. It might. We don't know that yet. Let me get to the other questions because we're going to get to those about recombination and Middle East camels in a second. And let me get to why are bats a common reservoir a little later. So some general properties of coronaviruses. Again, positive strand RNA virus, meaning the genome itself is infectious. If you just transfected in the genome of the virion, it would be infectious because it is going to direct the proteins that it needs to do everything else. No viral enzymes enter the cell with the virus. Replicates entirely in cytoplasm. The nucleus is entirely not involved. Again, the genome is a message. It has a genome-encoded poly A tail. It has a very large genome, so it's going to be larger than any other RNA virus. It's enveloped. Again, it's like a message. The viral RNA has a 5' cap. It actually has enzymes itself, which are going to put caps on its new message. It's going to make a large opening reading frame, and then it's going to process that large opening reading frame by viral proteases. So it's going to make a big polyprotein, and it is going to encode proteases, which will cleave that into a whole bunch of other proteins. And then it's going to make other proteins by transcription of subgenomic RNAs. Coronaviruses encode the largest RNA genomes. So on our left here, we have a bunch of different RNA viruses, and here we have sizes. And you can see most of them are much smaller. They kind of hit a limit of about 15,000 until you get to coronaviruses, where you get a quantum jump of coronaviruses being 30,000. There actually are some other ones of this order. The nidoviruses were up to 50,000. So this is unusual because something called Eigen's paradox, a version of Eigen's paradox this is from Manfred Eigen, is that RNA viruses can't get too big, because if they get too big, the mutation rate is going to, the frequency of mutations is going to be too large for the viruses to sustain replication. They're going to undergo something called error catastrophe, because they're going to, RNA viruses replicate at something called the error threshold. That is, they make as many mutations as they can to still survive. When you get a little bit above that threshold, you go into error catastrophe. Coronaviruses, their size should be over that size. So the RNA polymerases that make these RNA viruses make a mistake 1 every 10,000 base pairs. So these guys make 1 mistake every time they replicate. The coronaviruses have got around Eigen's paradox because they encode a proofreader. Sorry, so Eigen's paradox is that to get bigger, you have to have a proofreader. You can't have a proofreader until you get bigger. So the paradox is you got to do something to get there, but you can't do that thing because you can't get there. They actually have a proofreader. So they have the protein called NSP14, non-structural protein 14, is an RNA exonuclease. And it decreases the mutation rate about 10-fold. So the effective mutation rate for a coronavirus is about the same as you would expect for a genome that would be 3,000 base pairs. So it is making fewer mutations than you would have expected. So that's why when you look at the phylogenetic trees that are on next strain that we'll get to in a second, they're only seeing like 1 mutation per transmission versus a lot more, which you would have expected without this proofreader. So this proofreader is also going to be important to the next one. So some drugs against it, where the exonuclease, if you didn't have the exonuclease, it would be better. If you could inactivate the exonuclease, the drugs would work better. So doesn't coronaviruses disprove Eigen's paradox? Yeah, kind of. they do correlate it. So there are members of the order that don't have this exonuclease, and they are smaller. They are 15,000. There's a direct correlation between acquisition of the exonuclease and the ability to get bigger. So I guess it's still a paradox how it did it, though. So maybe that's not a paradox. So are the virions much larger to accommodate the larger genome? No, not really. They're packed. The nucleocapsid effectively packs them into as small a space. They're not actually that much bigger than other RNA viruses. They don't have enzymes into the virion as well. So negative strand RNA viruses have to put proteins in there, so that accommodates for the size differences as well. This one doesn't do that. So the message, the virion RNA is the message. The first thing that happens is you get this polyprotein made of replicase ORF1A and replicase ORF1B. The thing that separates ORF1A and ORF1B is a ribosomal frame shift. So because of an RNA sequence at the end of ORF1A, ORF1A is made as a big polyprotein. 80% of the time, the ribosome stops here. 20% of the time, because of the RNA structure, a pseudoknot in the RNA here, it slips, the ribosome slips back one, so it overrides the stop codon and makes a polyprotein consisting of both ORF1A and ORF1B. This is a huge protein, so about 800 kilodaltons. When this is made, so this is the polyprotein, there are two proteases that work in cis. So this part here, NSP3, encodes one protease, and it's responsible for cleaving the polyprotein at these sites shown in blue. And this one, NSP5, encodes a different protease, and it's responsible for making the cleavages shown in brown. So this is a viral protease that cleaves the viral polyprotein. These would be drug targets as well, but it's not so easy to make protease drug targets. The actual replicase, so the thing that's going to replicate the genome, is encoded by NSP12. So this structure of having these guys made more frequently these is that these are the enzymes down here that you need less of. So this is the actual enzyme, the RNA-dependent RNA polymerase. There's a cool protein here that's responsible for host mRNA degradation. So protecting it against the innate, degrading host mRNA so that future RNAs coming from the coronavirus get translated better. There's a helicase here. Lots of these other proteins, which I'm not going to mention, many of them are involved in RNA biology. So there are transferases, there are lots of RNA binding proteins. This is the exonuclease, this is an endonuclease. The drug that's in clinical trial, remdesivir, made by Gilead, works on the RNA-dependent RNA polymerase. So it's an adenosine prodrug. The prodrug is to get it into cells, and then there's a cleavage by cellular enzyme that cleaves out the adenosine analog. It binds to the NSP12 at the active site at +1 and prevents elongation. So this is acting similar to, for example, the HIV reverse transcriptase inhibitors, where you put a nucleoside analog and it prevents elongation. In the animal models, it's most effective when given early. So given late in disease, not so well, similar to the way the influenza drugs work when given early, don't work so well given later. We know that the target of this drug is the NSP12 because the way this is done in virology is you get resistance. So you derive resistance to your drug and you see where mutations that give you resistance, and the resistance maps to mutations in NSP12. This exonuclease is important because if you inactivate the exonuclease with mutations, you get increased activity of this drug. So the proofreader is actually limiting the activity of this drug. So a future strategy to make drugs like this more active would be to have a drug that works against the exonucleases as well as this drug. There are some other RNA-dependent RNA polymerase inhibitors which are in trials. There's one against the influenza PB1, which just was in the news yesterday. But those are the kind of things which would make good drug targets. So a question, does a host protease make the first cut? No, the host protease doesn't make the first cut. The host protease does make a cut of spike protein when it's actually getting in, and we'll cover that in a second. But the first cuts are done in cis, that is the polyprotein itself has protease activity. And then you make some cuts and then you get more protease activity. Let me get to the, Suzanne asked about chloroquine. Let me get to the chloroquine because that's a really interesting question that I'm going to show you in 2 slides when I go around the life cycle. Let me get to the rest of the way this genome is made. So we have our replicase proteins. The structural proteins are at this end. So we have stop codon here. So like many other RNA viruses, they separate temporally, making the viral genomes from making the virion proteins. So that's thought to give a time lag to help for the virus to help the virus hide from the immune system, from the adaptive immune system, by keeping its structural proteins till later when it has everything it needs to start making lots of them. So the structural proteins are here. Here's S, the spike protein. Here's E, that envelope protein. Here's N, that nucleocapsid. So they are not made from that initial translation, but each of these, they are made from a nested set of mRNAs. So here, each of these mRNAs is one of these open reading frames in a nested set going from, they're nested within here. In front of each of these mRNAs is this region called the TRS, transcription regulating sequence. And there's going to be a leader that's here. So this is going to be a form of splicing, but splicing done on the other strand. So each ORF has its own mRNA. Their abundance are not equal. Their abundance is going to be regulated by this process where the polymerase is going to jump around. So here's our genome. After you make some of those first replication proteins, you need to make an antigenome. So we're going to make an antigenome. This antigenome is in the negative strengths, and that's going to be a substrate to make more genomes. But the other thing that happens when you get a genome is the polymerase will come along, it'll get to one of these TRS sequences, and then instead of making an entire antigenome, it'll jump from this TRS to the leader over here. So it's going to make these subgenomic minus strands that are going to look like this, and these subgenomic minus strands are then going to be copied to make subgenomic mRNAs. So we're going to have this nested set of mRNAs that are now going to make one open reading frame at a time. So this is also the mechanism why the coronaviruses are going to recombine with each other. They have really high recombination rates because the polymerase is very processive, but when it gets to one of these sequences, it can jump from one genome to the next. So when it's jumping from this one to this one, it's making a subgenomic RNA. But if it was jumping from this one, oops, sorry. If it was jumping from this one to another full-length genome, it would recombine between the two. The thing that limits recombination between groups of coronaviruses is the sequence of the TRS. So clades of coronavirus that are close to each other will all recombine to each other. Those OC43 genotypes all recombine with each other. The origins of the SARS and SARS-2, they all look like recombinations in the bat reservoir. So it mixes and matches different genes that way. So let me get to the life cycle, and that's going to take care of Suzanne's question about the chloroquine. So first thing that happens, you get binding and fusion. This diagram is actually from a different coronavirus. It's from mouse hepatitis virus. So it's which is a model system because you have mice you can use. Also because it's not a human pathogen. This one and some other coronaviruses fuse directly to plasma membrane. SARS-1 and SARS-2 and many other coronaviruses do not fuse at the biomembrane. They bind here, then they get endocytosed. There's a cleavage event that occurs inside the endosome that allows fusion, that releases a fusion peptide that then allows the virus to fuse out of the endosome and into the cytoplasm. So similar to what happens in influenza, for example, and other RNA viruses that get endocytosed, they come out through the endosome. This cleavage inside the cell, similar to what happens with the Ebola virus, where there are host proteases, which are going to cleave the envelope protein, in this case S, which will allow it to fuse into the cytoplasm. So from the vesicles. So we're in the cytoplasm. We initially translate ORF1. We get transcription on virus-induced membranes. So the virus itself induces these double-stranded vesicles. It's thought that this is also a way to hide from the innate immune system. So everything is occurring inside these vesicles. We're going to have transcription of nested mRNAs, translation of the structural proteins, some genome replication, and then budding of the mRNA and the N protein back into ER vesicles. And it's going to bud out through the vesicles. So now the question about chloroquine derivatives, there have been some tissue culture experiments and some limited clinical trial. No, clinical trial is not a good word for it. Giving chloroquine derivatives to people with this virus, I don't think there were any controls, that showed promising. So chloroquine is changing the pH of the vesicles. So that could work on the way in, so preventing fusion from those vesicles. Possibly that's changing something in the replication within this double membrane vesicles. And possible that's important for this budding out. It's really not clear which of those is true, but that could be true. So Susanna, does that mean if I isolate mRNA from virus and then the qPCR, that qPCR products are infectious? No, people are not doing qPCR for the whole genome. They're doing qPCR for just little parts of it. So that is not infectious. If you isolated RNA from a virion, it almost certainly you would have some RNase activity around someplace. So when I say that the RNA in the virion is infectious, that's usually people synthesizing in vitro full-length RNA and transfecting it. You probably could not get that from a virion, but you might be able to. I understand you're saying everything's happening in the cytoplasm, not the nucleus. Yes, everything happens inside the cytoplasm. There's no role of the nucleus here. Totally RNA-based, no need for any DNA machinery. So SARS-1. So this epidemic was in 2003 in southern China. Guangzhou is the largest. It's going to be important here for the epicenter of this. And there's an important date in this epidemic, February 21st, 2003. That's the date when a physician who was treating patients in Guangzhou went to Hong Kong to attend a medical conference. He spent one night in Hong Kong, and the rest of the epidemic followed from that one event. This can be characterized into 3 phases. So there was a sporadic phase here where there were cases they were spaced apart and they were fewer. So this is called the early phase. So this is, again, happening in southern China. The epidemic phase starting around here when these cases are increasing, and then the resolution phase here. February 21st is right here, so kind of in the border between the epidemic and the resolution. I'm going to get back to some characteristics of sequences from these different phases in a second. But this is what happened on February 21st, 2003. So patient A went from Guangzhou, spent a single night in the Metropole Hotel in Hong Kong. He vomited on the mat in front of his hotel room. The next day, or actually that evening, I believe, he went to a hospital in Hong Kong. Patient J and patient H went to 2 other hospitals in Hong Kong. Those 3 were responsible for 134 healthcare workers being infected and 156 close contacts of those healthcare workers. Patient B went to Vietnam, seeded this outbreak. Patients C, D, and E went to Singapore. Patients F and G went to Canada. Patient I went to the US, and patient K went to Ireland. All of these guests were on the 9th floor, the same floor as patient A, except for G was on the 14th floor, and K was on the 11th floor. So this brings up something called superspreading, which we definitely saw with the SARS coronavirus, and we may see with the SARS-2 coronavirus. So the R0 that we calculate, so how many people will be infected from one infected person, that's a simplified way of talking about the R0, is an average, but it's actually not evenly distributed. So there are super spreading. It's not known why there was super spreading, but we've seen that here. So in South Korea, they had patients 1 through 30, where there was not much untoward spread. Patient 31 went to a church in South Korea. 80% of all subsequent infections in South Korea are derived from patient 31. Again, it's actually not known really what's responsible for super spreading. So this was the epidemic, 2002, 2003. Most places around the world, mostly in Asia. So the reason that many places in Asia are prepared for this new one is that they had the most experience with the last one. So Singapore, Vietnam, Hong Kong, Taiwan. 8,000 cases worldwide, cases in Canada, no apparent deaths in the US. 25% of those required intensive care. So that is going to be kind of similar to what we're seeing here. So lots of people requiring care in the hospital. In this case, the case fatality rate was about 10%, 774 deaths out of 8,000 cases. So that's a high number. If we look at sequences from those early, late, and middle phases, so here's a phylogenetic tree of the early ones, the middle ones, and the late ones. We see decreasing diversity as we get further on in the epidemic. If we look at the DN to DS ratio, so the number of non-synonymous mutations to synonymous mutations, so when this number is bigger, it means that there is positive selection going on. If this number is low, it means that there is negative selection going on. So a large number will often mean that there is selection for the virus to find a fitness peak, whereas a low number means that it has found its fitness peak. So the interpretation of this is that, for example, if we look in spike, and if we look at ORF1A, early in the epidemic, this virus was not completely fit for humans. Late in the epidemic, it had found a fitness peak. So it had become adapted to humans by the late phase. So late phase meaning the phase in China where then it went to the world. So once it went to the rest of the world, it was already at its fitness peak. We are not seeing the same thing with the SARS coronavirus 2. That is, we're not seeing this kind of diversity, meaning that there were cases that previous cases that we have not sequenced where it was not yet adapted to humans. But the sequences we're seeing now, it looks like it has already been adapted to humans by the time we are sequencing. Sorry, what does tree tell you? That's from class. I just told you what that was. SARS-1 appeared first in a wet market in Guangdong. These are pictures of raccoon dogs. Early on, it was noted that pomp civets and raccoon dogs from the market were infected. Initial infections in Guangdong were in restaurant workers who prepared exotic meats. Sequencing from animals in the market showed that most of the pomp civets and most of the raccoon dogs in the market were SARS positive, SARS coronavirus positive. The viruses from the pomp civets and the raccoon dogs matched the early cases of the pandemic. However, none of the 1,000 pomp civets, the wild pomp civets in the surrounding provinces were positive, meaning the pomp civets became infected within the market. They are not the original reservoir. They could be the proximal reservoir, but they are not the actual original reservoir. The original reservoir is the horseshoe bat. So this is what they look like. They're small. This is somebody's thumb. They're distributed throughout China. They are actually distributed. The viruses we find from bats are not unique to China. They're also found in Europe and in Africa. So here's another phylogenetic tree. Here's this close relationship between SARS coronavirus and humans, SARS coronavirus and a civet. So very, very close. So much closer than anything we're finding with coronavirus 2. So in terms of an animal reservoir, here are the bat sequences, which are the outgroups, close outgroups of that. The spike protein of this is ACE2. This is the same, sorry, the spike protein used this as the receptor. This is the same receptor as the SARS coronavirus 2. It was originally thought that the bat isolates use a different receptor, but since then, in more recent years, they have found bat SARS-like viruses, or in the same clade, that actually use the ACE receptor, ACE2 receptor, and can use human ACE2 receptors. So the new hypothesis is that there actually wasn't, it didn't have to go from bats to civets to humans. Civet cats were infected at the same time as humans, and transfer could have come either directly from bats or from an intermediate host, but it probably did not have to adapt in civet cats. So there was a lot of stuff earlier on in this epidemic about snakes and then pangolins, which are these armored anteaters, being the source. They are, the snake thing is all wrong, but the pangolin, that sequence is too far away. And it does not actually have to be something that was in the seafood market in Wuhan. We actually know there may have been cases that pre-existed that seafood market outbreak. The seafood market, the amplifier in the seafood market may well have been humans and not anything else that those humans were eating. So the end of that epidemic, the R0 was about 2 to 3, but some super spreaders. Difference between SARS-1 and SARS-2. With SARS-2, people were most infectious when they were already sick. Most of the infections were occurring in the hospital. So someone was severely ill, went to the hospital. The peak viral loads were actually during that time. So most of the infections were actually occurring in the hospital. So between the patients and the healthcare workers, or between those patients and other patients in the hospital. But most of the infections were occurring actually in the hospitals. So the end of this epidemic was because of those things. So barrier nursing, which is what it sounds like. So preventing infections from patients to healthcare workers, preventing infections from patients to other patients. Quarantine, so quarantines worked well because you knew who was spreading because they were already sick. Again, travel restrictions also reduce that spread. So this is one we were able to control. We don't have some of those same luxuries with this one, because in this one, in a slide I'll get to later, people are transmitting, viral loads precede symptoms, not after symptoms. The last case of this current epidemic was in Taiwan, 2003. Again, this idea that it goes away in the summer. We don't have some of the things that could have happened. It's not going to go away in June. The virus did reappear in 4 sporadic cases in Guangdong, 3 laboratory accidents that were not using BSL-3 protocols. In January 2004, they culled the market of all the pomp civets and the raccoon dogs, and there have been no SARS-1 since then. The pathology of SARS-1, the pathology of SARS-2 are very similar to each other. Infection of the pneumocytes in the alveolar epithelium, followed by infection of macrophages. Virus is higher in the lower respiratory tract than the upper. With SARS-2, there may be more virus in the upper respiratory tract than there was for SARS-1, which may account for the increased transmission of SARS-2 versus SARS-1. There was acute damage to the lungs. This may be partially immune mediated. So with one of the treatments being done is a humanized antibody to the receptor IL-6. IL-6 is a marker of immune stimulation. So an antibody to the receptor appears to is being tried in clinical trials. So some of the damage is immune mediated. Someone asked about the intestine. There is virus replication in the intestine because the receptor is expressed in intestinal cells. This does seem to be a thing of other coronaviruses as well. It is not clear that there is any transmission from the fecal route though, but there is virus being secreted. In this case, viral load peaked 10 days after infection, which is going to be different. Virus is cleared within 2 weeks, but lung damage is so severe that many deaths are occurring after that. As most patients actually died 2 weeks after infection, after the viral loads had already gone down because of the viral damage that had been done earlier. So that's why the case fatality rates that were being calculated earlier, for example, from the first cruise ship, they were off because they were doing it too early. Those people who would pass away are going to pass away later on. MERS was another one. So MERS, the first patient was in London. He originally traveled from Saudi Arabia. It has a bat origin here. This one is not endemic in the human population. Here are MERS outbreaks here. So each of these peaks is a different outbreak. It also has a high case fatality rate, in this case 34%, uses a different receptor from SARS. But each outbreak is a different transmission from camels. And then there is a limited transmission from human to human, and then that dies down. And then each new one is a new index case. So this one does not human-human transmit, unlike the SARS-2. So here's why they call it SARS-CoV-2. It's grouping clearly within this clade. Here's SARS-CoV-1. This is a bat sequence. Here's SARS-CoV-2. This is a bat sequence. The other ones are in different clades. This is the way that viruses get named. So it is definitely in this clade. Erica, can you speak about mechanisms of the heart defects? Okay, someone asked that last time, and I should have looked that up more because I do not know that. I know that there is reports of that, and it is important, and I don't know anything about it, unfortunately. Do we ask about NSAIDs and COVID-2? There's no data on that. There was some speculative report on maybe it had an effect because of its effect on the ACE2 receptor. That's all speculation. None of that is, nobody really knows. If you have not looked at Next Strain, you should definitely look at Next Strain from Trevor Bedford's group. super useful, lots of good information there. You can see how they make inferences from phylogenetic trees to pandemics. These next slides are ones are taken from screenshots from the Croy lectures that hopefully you got that link to those. They're really wonderful talks. Oh, someone asked about ACE inhibitors, more susceptible. There's a lot of discussion about that. There's nothing known about it. I think people are recommending that there may be a switch, but there's actually no data on ACE inhibitors. So here are case fatality rates. So here are fraction of cases. This is from 44,000 cases in China. This is from a wonderful talk at Croy. So here are fraction of total cases, and here are the case fatality rates. So this is actually mirroring what you would see in influenza, but 10 times higher for everybody. So especially high case fatality rates with each decade over 50 years old. One difference from influenza is we are not seeing deaths in the 0 to 2-year-olds. So 0 to 9, not seeing that. Starting, this doesn't mean there's no mortality under here, but just in a relative effects. In each way you look at it, if you can put this on a log scale, it's 10 times higher than it is with influenza. So that gets to be really dramatic with people over 80. So this is why virus going into nursing homes, assisted living homes, those are worst case scenarios. So here's fraction of cases. So this looks like there aren't cases in younger people, but this is a biased set because this set is biased by people who were showing symptoms and being tested. If you look at it a different way, so in this case, this was a study from China where they took people who were showing symptoms, so in the hospital, and then they went back and traced all their contacts. So for each person, now they're looking at contacts. So 5 to maybe 10 people of their contacts, often within a family, and they're going to ask the attack rate with each age group. So now this is not focused on who was sick, but who had contact with someone who was sick. They're going to ask the proportion which is severe and the portion without fever. So this is super important because the attack rate, so the percentage of people who are infected, is actually age independent. So younger people are still being infected. So here is one of the major reasons why schools are being closed all over. So it's not that children are getting sick. So here you look at proportion severe, where this number is super low with children. Proportion without fever, so no symptoms basically, is really high in children. So here and here. But children may still be infected. Children may still be able to transmit. So keeping children away from their grandparents is super important. Preventing spread within the community may be very important with children because, again, the children are not symptomatic. This mirrors other primary coronavirus infections where children do not be much affected. Children are affected, but mostly asymptomatically. Again, in this, if you look in a non-biased way by context, you see much more severe, the portion severe goes up with increasing age. This is from those same things. So the critical people is right here, 5%. Mild infections are 80%. This is the important number here. Severe infections around 14%. So this is the important thing. So rather than focusing on who is going to survive, this is the thing that risks to overwhelm the healthcare system. If 14% of the people need to go to the intensive care unit, this is going to be a big number. So we're multiplying percentages by big numbers, which is going to be actually a big number. Symptoms of the coronavirus are mostly non-specific. So it looks like influenza. Influenza is still circulating in most of the United States, definitely circulating in Washington. Many people with fevers do not have coronavirus, they have influenza or other respiratory viruses which are co-circulating. So fever, dry coughs, fatigue. As you'll see, runny noses are not on here. If you have the sniffles, that's an upper respiratory tract, it's something else. Time from exposure to symptoms, this is important, 5 to 6 days. So meaning that people can't transmit before they start being sick. People who were exposed to someone who subsequently became sick, it's already too late. So exposure could have occurred from that. So all of this social distancing is because of these things, because we need to keep people from people and not just sick people from people. Asymptomatic infections actually is known. We do need serology. So these numbers are all biased by, we know who's tested, we don't know who has been infected. So until we start looking retrospectively at who was infected by who was antibody positive, we won't actually know a lot of these numbers. Virus shedding is highest earlier in the course of the disease. That's the other thing that's dangerous. Virus shedding can occur before symptoms, and it continues throughout symptoms. So many of you have seen this curve in social media. This is called flattening the curve. So here's the number of cases. Here is time since the outbreak. This is what happens if we don't do any social distancing. So this curve and this curve may have the same area under the curve, so same number of people infected. But if we go this route, this line here is the capacity of the healthcare system. It will overwhelm the healthcare system. If we go this route, we may have the same number of infections, but it will be, the healthcare system can take care of those people who with severe symptoms. Again, this is drawn as an even distribution. It's not going to be an even distribution because of the seasonality. Lower in the summer, there is a risk that it comes back in October. There is a risk that it becomes endemic in the human population, but there will then be hopefully drugs, and there may be vaccines. Here are cases from as of Tuesday. Sorry, this is an incorrect slide. It says COVID cases. They're confusing disease and virus. This is number of virus positive people. Again, this is super biased by how many tests are being done. So this is test positive. So here's China. It's going down in China. The last cases are, the Chinese cases are all being imported and not domestic. Since then, there are more cases being reported each day outside of China. So this is not a cumulative graph. This is cases reported per day. So the world now has more cases than China. Some places this is going down. If we look in South Korea, these light oranges, most other places it's going up. So until these numbers start going down, the end is not in sight. So we don't know if what we're doing is working until these numbers start to go down. Questions? That was everything I think I had to say. So, other questions? If the curve is flat with social distance, will there be fewer cases? That is hard to say. So it has been said that we may be beyond containment and we are now in mitigation. So this flattening of the curve is all about mitigation. So we may have the same number of cases, but they may be spread out more. They may be spread out enough that there is immunity in the population that also will take into account the R0, the R naught of driving it down to 0. It could be that it becomes endemic in the population. We may see the same number of cases, but they will be more spread out. Why is there a truncated day? Oh, I forgot to talk about this day. This day is an artifact. This day, this one day, China changed the way they reported. And instead of reporting by the virus positive, qPCR, they started reporting by symptoms. This is indication that the number of people actually positive are much larger than actually reported. So this was some imaging tests that they were doing on lungs. They decided that was a bad idea because it's less specific, so they only did that one day. Sort of testing the CDC uses. The CDC is using, everyone's using a qPCR test. qPCR test, so primers and an internal probe. The test is done from the upper airways, so putting swabs in the back of the throat and the nasal cavity. The virus is mostly replicating in the lower respiratory tract, so that some of the false positives is that it's not the actual source. The amount going out through the upper respiratory tract can change over time. So that can be also why someone is negative then positive when they actually were positive all the way around. There are limits in detection. There are things about false positives because it is a qPCR. Serology tests are being developed. Serology tests are going to be super important. They are not available yet, but people are working on that. There are ELISA tests. There are more rapid tests for antibodies. So possibly looking at IgM very early in disease will be very useful. Definitely looking at IgGs later on, asking about titers, asking about what amount of titer is necessary for protection is very important. Convalescent serum as a treatment, I would say could be, but there'll probably be other antibody-based things going on. There are humanized antibodies that may work. Those kind of things are pretty intensive, probably not going to be used at a large scale. Nobody asked about vaccines. Let me talk about vaccines. So there are promises about vaccines coming on. That could be great. That's not a guarantee. So there are not existing human coronavirus vaccines. It is possible that a vaccine with the right adjuvant will induce amount of neutralizing antibody that is protective. Until we actually have a vaccine that shows that, we won't know actually is it possible or actually is it how hard it is. Vaccines are tricky things to do. You cannot promise that a vaccine will be ready in a certain time date. It might be, it's hard to predict. Neil asked about models, about rebound and social distancing relaxed. Yes, that's a problem. That all is going to depend on the amount of immunity in the population that's going to occur next fall. It's going to depend on what happens in the southern hemisphere during their winter. It's going to depend on how good a vaccine is and how many people will take the vaccine. Models are fine for things, but until we actually have data on those kind of things, I don't think we can model. So again, the idea is, will we be able to eliminate this or will it become endemic? I'm going to emphasize that there are endemic coronaviruses that came from animal reservoirs that are now circulating in the human population. It is possible that what this will do, it will become a childhood disease. Children not being infected, but not having severe symptoms. Everybody will get it in childhood. There'll be protection in adults. That is a thing that could happen. That is, it could be a pandemic for the next year or the next year or two, and then become endemic and not be a severe problem anymore. One of the vaccines being tested is an mRNA vaccine. Have there been any successful mRNA vaccines? I don't know the answer to that. Does anyone else know the answer to that? Whether there's a successful mRNA vaccine? I mean, similarly, people are doing DNA-based vaccines, and if there's an example of that, that example is probably coming from the veterinary market. how feasible is the drug targeting NSPOR 14 proofreader? So NSPOR 14 is a dual specific protein. It actually has methyltransferase activity and this exonuclease activity. So it's going to have some unique structures. I am not enough of a structure person to say how different it is from existing exonucleases. Again, it is a cytoplasmic protein, so that's a good thing. Are there obvious reasons to avoid developing drugs targeting this protein with the idea that you would not do it alone? So you don't really want to, there are ideas that if you increase the mutation rate, that alone causes viral catastrophe, but increasing the mutation rate in the context of improving other drugs would be the goal, so not doing it alone. Let's see, questions have slowed down, so hopefully that means that I answered everyone's questions, but thanks for listening, and I am going to, I think I forgot to tape this. I'm going to tape some version, probably next time. If I forgot to hit the tape, which I may have done, I'm going to be able to send the slides to people. Hopefully you thought this was useful. Someone says record at the top. Yeah, I knew that, but I think I forgot to hit the button. Anyway, I'm doing another version of this talk a little bit later, but hopefully you thought it was useful. Feel free to send me an email and tell me how I should change stuff or what you really wanted to know and I didn't answer. But, oh, someone says it's flashing recording. So I guess I did record it. Okay. So I'll try to distribute that recording. Thank you very much.